Today, I'm delighted to be joined by Dr. Pan, and he's the co-founder and chief technical officer of KaloCyte in our series of interviews with KaloCyte founders and leaders. We're going to dig into his background, the KaloCyte story, and everything. So, Dr. Pan, thank you so much for joining me.
Well, thank you, Glenn. Thank you for having me. It's great to be here.
Yeah. I wanted to kind of first your story, an incredible story around the science. You know, you're really involved in nanotechnology and that inspired based applications. For somebody like me who's very ignorant to that world, when I think nanotechnology, I go to Marvel and think of Ant-Man and Iron Man and that world. I can't imagine they were your inspirations when you were younger, started out in this field. So, if you could just give a little bit of a background in terms of, what sparked your interest in, engineering biology, if you like.
Yeah, that's a great question. So I chose nanomedicine because it kind of sits at the intersection of imagination, as you were saying, and impact, right?
Hmm.
So it really allows me to engineer at the tiniest scale while solving, some of the biggest clinical problems from early diagnostic to smarter therapies. So for me, I think it's kind of a perfect way to combine curiosity with real world application. But I mean, it did not really start that way. So my training really began in chemistry, in material science, but I was always fascinated by the idea that biology is probably the most sophisticated engineering system that one could think of, right? So, with that kind of promise, I call myself a bioengineer and a contaminated chemist. Because we apply chemistry and material science, tons of biology, but then engineering mindset to solve biomedical problems. For this particular problem, we were trying to mimic Mother Nature, right? So red blood cells, for example, it's uh, it's uh, it's really extraordinary if you think about it, right? So millions of cells that are flowing through our blood vessels, and each of them are carrying oxygens. And those have been optimized for circulation, for gas exchange, for immune compatibility over millions of years, right? So that's how the evolution happened. So what kind of drew me to this kind of topic was the realization that we really don't really have to replace the nature. We just kind of have to mimic the nature and the properties that nature already created, and by learning from it. So if we understand the structure, function, relationships, so that means we're bringing the structure, that's chemistry, the function is basically biology, and then we're applying engineering principles to put everything together. We can design materials that will emulate biological intelligence that would be next generation.
Yeah.
So it's about decoding the biological design principles and rebuilding them with precision and scalability. So it was not that I started as a nanomedicine scientist, but I really started as a chemist. But then my interest evolved as my career progressed.
Yeah. And the industry, doesn't it? The world progresses is like no one could imagine where we are now in terms of understanding capability and what science can do, you know,
Oh
5 years,
yes.
let
Oh,
alone 15 years ago.
absolutely. Because the technological tools, I think, evolved so much, right? So just think about, one classical example is X-ray imaging, right? So 1895, William Ronson discovered X-ray, right? So we know that famous first X-ray image of his wife's left
Yeah.
hand, showing the wedding ring. Fast forward 100 years, now we have, all in that picture, all we could see is some um dark, the bones and no soft tissue, nothing. But fast forward, if we look at some of the emerging CT images with the technologies such as photon counting imaging and others, we are seeing everything in color, right? So technological development, and I'm just giving you one example, the
And
technological developments that happened, it made a major impact in today's medicine,
yeah,
and
it
it's
is.
continued to do, and it's continued to do that.
Yeah,
It will.
it's amazing. I think coming on to how KaloCyte came about, and we've already had, I already had the pleasure of speaking to Dr. Alan Doctor, and he described you two kind of getting together, not as like a chance meeting, but it was kind of two worlds that didn't realize that there could have been something possible because of individual work that was going on. And then it was like, ah, hold on, there's a real opportunity here. And it kind of, you like solved each other's problems. Is that what happened in that sense?
I think he's right. So it was. I think we could write a book about
Let's
this.
get to human trials first and then we'll do the book.
I think so we were both at Washington University in St. Louis School of Medicine, and we did not know each other. And my lab was trying to develop a nanoparticle for imaging MRI with as an MRI contrast agent. And the particles that we developed, and again, I think with the concept and with the idea that mimicking Mother Nature, that the idea was that the nature solved some of these problems so elegantly that when it flows, so for example, red blood cells, when it flows through the blood vessels, the hemodynamic properties are um, perfectly optimized for traveling within the blood vessels. So the idea was, again, to learn from that solution and engineer it for the realities of modern medicine. And the MRI contrast agent was one of these ideas that how about we make a nanoparticle that looks like a toroid or donut shaped, and then we see whether it can actually carry some contrast agents for lighting up some of the diseased tissues. So we made those particles, and when we looked them under um, electron microscope, we found out that they, it was kind of a serendipitous that they look pretty similar to red blood cells, but it was much smaller because these particles were, somewhere around 100 nanometer in size. And at that point, I think, I started wondering whether these particles can also carry hemoglobin, and we can utilize them for delivering oxygen to the deprived tissues. And through a mutual collaborator, we met from Alan Doctor, and I had never worked in transfusion medicine, and he had never worked in nanomedicine. So when we sat together, and we realized that there is an enormous potential for us to collaborate, and we bring complementary expertise, and then that's how it started, and it's been more than 12 years. I think we've been collaborating together. We started KaloCyte, and I think this is probably one of the longest running collaborative relationships that I've ever had, or my lab involved with.
Yeah. And it's
So
actually
it's the
still
story is
ongoing.
fascinating.
Yeah. Yeah. Well, from that point of where you were talking about um, bringing kind of two ways of thinking and two disciplines kind of together to create this solution, how does then it go from that, you know, from like the bench when you're thinking about it to the reality? And what are those what are the challenges, if you like engineering this this technology and designing it to be manufactured? What have kind of been those challenges over the 12 years? And I can imagine they still exist now, to be honest.
Yeah, yeah. So this is again a great question. So it's understanding that that science is, good science is not enough, right? And I learned it very early in my career, that and that was, I think it was kind of a my pivotal moment, that realizing that brilliant science, a lot of labs do brilliant science, but that's probably not enough. So I have had technologies that worked beautifully in the lab, but the translation required different kinds of things. For example, understanding the clinical problems, the complex biology behind it, manufacturability, regulatory awareness, looking into the cost, and all those kind of things. So, it's about the mindset that you need to have, and that innovation can happen, or must be engineered from day one, that you're really trying to think about. And that's how I think, when Alan and I we started KaloCyte, the philosophy was kind of central, that we're not really working for this is just a publish or perish type of work, but this is something, we're building a life-saving therapeutic platform that sooner or later will be injected in human. And so that was the primary focus from day one, that it requires a whole lot of understanding about these things, that we typically in basic science lab we don't really think about from day one.
Mm, okay. It is fascinating. Like you say, there's this challenge of like focusing on the science, but if you want to achieve anything, you've got to go beyond the science, haven't you? Like you say, from a safety point of view. And when we spoke with Elaine, your CEO and president, is culture and values as you're growing the
Correct.
team, and what your approach is key to it. It's not just the science.
Absolutely. And so in the beginning, when I started my faculty career, my entire energy kind of went into starting the lab, publishing, securing grants, like most of the new faculties they do, right? Keeping tenure in mind, because I had to build the credibility and independence in my field, right? So I was not really sitting there thinking like, how do I start a company? That's a very, very wrong approach, right? So my and then slowly my mindset started thinking, it was more closer to, how can I really apply this science to solve important problems? And then entrepreneurial opportunities kind of emerged naturally, when the research pointed to the solution that had real world value. So, again, I think pursuing this approach is not really, and this is really for newer generation of scientists, that, if you're pursuing translation, it really does not distract you from doing absolutely great science. It does not distract you from getting tenure or other things. In fact, it will strengthen someone's academic career, because what you're doing brings tremendous impact to the field. And the translational work, for me at least, it brought enormous visibility to my lab. So I started 5 companies so far, and I did not start with entrepreneurship in my mind. This is kind of my North Star, but started with curiosity, and
Mm,
keeping in mind that if the technologies that we're developing, the science that we're doing, if we can really translate it for human use. So
yeah.
it's a natural extension of my academic mission.
Yeah, that's
Is
some
that
great advice as well to anyone who's, early on in their career, or do you know what, even sometimes who are quite far in their career, sometimes just need that moment of realization, don't they?
That's right.
Uh,
That's right. That's right. But I mean, there is a thrill of this translation. There is a uniquely exciting about watching an idea move from a bench experiment to a product in someone's hand. And it's absolutely messy. If you talk to Elaine, if you talk to Alan, you will know, because we are we're living and breathing this life every day for the last 10 years or so. It's absolutely messy, it's uncertain, but at the same time it's deeply rewarding.
Yeah.
So
That's the exact question I asked Alan as well, is like what kind of keeps you energized. And I think this is a life thing more than anything. It's enjoying the journey and not getting obsessed with the end goal, thinking that's the reward. The journey is the reward. So that's the part that you
it's
have to embrace.
absolutely, absolutely. And it's because the next problem always calls, right? So my lab is, and this is a little bit of I don't want to completely derail our conversation, but this is I think this is important to mention that it's always the next problem that calls, right? So my lab is very multidisciplinary, and the world is full of unmet clinical needs. So once you have had a test of solving one problem, then you start seeing opportunities other places, and that will drive you to make a difference, and then that keeps me coming back. For example, I can give you one very simple example that we are now fascinated by, one vision that I have, that how can we democratize women's health. And we're looking at menstrual effluent, period blood, which has long been considered as a biological waste product, right?
Mhm.
So the idea is that how can we take that and then get information about someone's health, and how can we really, that person can collect that and test it in the convenience of their home, and test it to understand how their bodies are doing, for example, about endometriosis, or cervical cancer, or PCOS, or all those kind of women's health.
Yeah.
So and what we have seen, that we have published a few papers, and then there was a lot of early industry involvement, and what we have seen is a tremendous amount of interest in this technology
Yeah.
that we are currently developing. So just wanted to let you know that this is the three love translation is real, and it's
Yeah.
always about the people and not just the technology.
Yeah, that's kind of perfectly segued to my next question. And you mentioned having multidiscipline teams at KaloCyte, and when we spoke to Elaine, she spoke about the culture where no idea is a bad idea. Everyone kind of gets together, they share those problems. So can you give some insight in terms of like how those ideas flow through KaloCyte and the teams, and where that innovation comes from in terms of like the culture and, like you say, that cross collaboration?
Yeah, and yeah, that's again a great question. So the question really is, how can we really um, engineer a bio-inspired red blood cells, right? And trust me that we have had harder fourth generation of particles. Okay, so we started working on one design, and then moved on to many different iterations and versions of it, because the idea began with a fundamental question, that what makes a red blood cell so effective, right? So we can think about high oxygen carrying capacity. It is deformable when it's flowing through the capillaries, and in its stability in circulation. So the idea was, from the very beginning, that rather than using a free hemoglobin, which is very toxic, we will engineer a stabilized hemoglobin in a protective shell that mimics the structural and functional architecture of a red blood cell. But the question is that how can the design integrate the controlled oxygen affinity tuning? Like, for example, like how can it respond to the tissues that are oxygen deprived? We're also thinking about structural stabilization, right? So that it does not really fall apart when it is injected inside our body. So, and then we were thinking about the surface chemistry that minimizes the immune activation and a manufacturable synthetic architecture. So, all these are bio-inspired, but we really need to make it intentionally simplified for
Mm.
scale, robustness, and all these things. So now, when I say all these things, you can see that there are already, we're talking about material scientists that are bringing the concept about making these particles, making the hemoglobin stabilized in a protective shell. We're talking about um, red blood cell physiology, which will teach us how to really gain the controlled oxygen affinity by creating a protective shell, or at least giving us some ideas about creating this protective shell. And then we're talking about a manufacturable synthetic architecture, right? So that means we need tons of industry experience to produce these materials in a large scale without any trouble. And also remember that our goal is to make lyophilizable particles so that those particles can be resuspended and be administered at the point of injury, right? And that's a tremendous, it sounds really great, but that brings a lot of complexity to it. So it's just not a one person job that we're really trying to do, but it's multiple areas that are combined together to achieve this technical breakthrough.
Even you didn't even mention there all the things around it, all the regulatory safety work that has to go on as well. That you have
Correct.
to get past all those milestones, isn't it? So it's
Absolutely.
that's
Absolutely.
before you
So
even get into the commercial aspects when hopefully we get to that point.
yes,
Yeah.
absolutely. I mean, you have to maintain really batch to batch consistency. You
Mm.
have to prevent the hemoglobin oxidation at scale because those are all very, very critical. You have to ensure purity, control the particle size distribution under GMP. So as we approach the scale, the design constraint, it can come in if you're not thinking about it from day one. So the process engineering chemistry are really co-developed, not sequentially, and then biology comes in, and understanding of biology comes in to help the process.
Yeah.
So designing for manufacturability, I think it's the emphasis should be placed very early on.
Yeah. Yeah. It's incredible what all of it yourself and your team's doing. I think it's so exciting to see, what the
Yes.
future months and years holds for KaloCyte. Moving away from KaloCyte, and anyone watching and listening, they don't have to hold Dr. Pan to this, but and we spoke about the speed of innovation and development. If you look to the future of, bioengineering or just generally the red blood cell development and what's in that space, how do you see the future of this world? What can we maybe expect to see in the near or distant future when it comes to this? Have you got any predictions? You must think about it a lot.
Well, yeah, I mean, absolutely. I think this is something that the technologies, as you know, they're shaping the future of if I may say, bioengineering, right? So that's an interdisciplinary field where we really combine biology, chemistry, engineering all together. So I really think that moving forward, artificial intelligence will really drive some of this molecular design that we are doing. When we started AI, the concept of AI was not really available at that time, or at least not with the term AI.
Yeah.
And I know I think the machine learning and best tools will really drive these molecular designs, advanced bioprocessing, and continuous manufacturing processes. Also, bioengineering is really moving more from descriptive biology to programmable biology, right? So we can really think about even more advanced rithomar if you are to design right now, because that will probably open up possibilities around cell-free synthetic biology-based platform or precision protein engineering, and then we can combine it with advanced bioprocessing and continuous manufacturing. So I think this is where the field is moving towards, and artificial intelligence will play a big role in those kind of activities.
Yeah, it
And
feels.
yeah, and if I may say that the capabilities that will define the next decade or so would be the platform thinking. If you're thinking about platform over a
Mm,
single product, I think, and thinking about from a real world deployability, like shelf life and whether it's field ready or not, and science will be driven by a regulatory aware design. So from day one, the design will be regulatory. We need to think about regulatory aspects of it. So
yeah.
integration of biology with material science and computational technologies would happen very naturally.
Especially with that regulatory thing, because we're weighing up kind of two things here, aren't we? We're like national regulatory bodies specific, you know, the FDA and in the science world, but then there's wider discussions, and you mentioned it's specific to the use of AI in general across
Hmm.
all industries, isn't it? So
Correct.
like those worlds are going. Well, they probably are already. They are clashing, and we've got to work out how they all live together a little bit.
Well, we have to. We really cannot avoid that because it's already here.
Yeah,
And it's our responsibility to use AI in a responsible manner. And but it is so powerful that it will help shape the future of bioengineering, including oxygen therapeutics.
yeah, yeah. It's really exciting, and yeah, it's great to get the background, the journey you've been on, and so insightful in terms of kind of what's been happening. So I just wanted to say thank you very much for joining. And for those who this might be the first episode that we've looked at, please make sure and go and hunt out the other interviews with people we mentioned in this episode, and Dr. Elaine Haynes, who work alongside Dr. Pan. But just want to take this opportunity to say thank you very much, Dr. Pan, and good luck with hitting all the future milestones for KaloCyte.
Thank you so very much, Glenn. Very nice talking to you, and thank you for the time.